Hello again and welcome to Chapter 21, DNA and Biotechnology. So this is our third topic on DNA, our third chapter. And so we've hit cell division, we've hit genetics, and now we're going to look at gene expression, genetic engineering, and genomics in DNA and Biotechnology. Okay, so we've looked a little bit about genetics in the previous chapters, but overall, our DNA is 4-letter languages. So we've got 4 letters, A, T, C, G. And that order of those 4 codes for RNA, and then that's modified, and it's then made into proteins using amino acids. So DNA's language is a little bit different than RNA, but it's also 4 letters. There, instead of a T, RNA has a U. And this table is showing how our RNA codes for different proteins, for different amino acids. Basically, it's 3 letters in a row, and that is the code for one amino acid, and that's called the codon. So the codon, like for example, AUG, codes for AUG, methionine right here. So that's what all of our proteins start with. And then you'll read the next codon, match up the next amino acid, and you'll make a longer and longer protein. And then it'll start to go into those different shapes, the helices, the sheets, and so on, to make a protein. Pretty exciting. So gene expression is taking that DNA and making proteins, expressing the gene, right? It's the central dogma of biology. So reading genes, making proteins. And there are 2 parts, transcription and translation. So the transcription portion is the DNA to RNA. Those are both in the same language of nucleic acids. So it's basically rewriting the same thing in the same language. Whereas translation is the RNA to proteins. So there, we're changing the language from nucleic acids to amino acids. And we'll do a summary slide where we go through all of this. Okay. We'll start out talking about transcription. So transcription, we take that double helixed DNA and we open it up. We've got a couple enzymes that help out there. And then the enzyme RNA polymerase starts reading the DNA code and then putting on a single-stranded RNA that matches it. And so, for example, let's say our DNA starts out, we're looking at it, the code is CGTAG. So our RNA matchup, C always matches with G, and then A, and now we don't have T in RNA. They must not be British. Oh. Instead we have a U and a C, so they match up. Okay, great. And then translation is making proteins. So that occurs at, where did transcription take place? Where's DNA? The nucleus. Whereas protein synthesis, remember, that happens at ribosomes. There are some ribosomes free-floating in our cytoplasm, and there are some attached to the rough endoplasmic reticulum. And where the protein is going is going to determine which ribosomes it goes to. But basically, you've got your ribosomes, and they've got 2 parts, this kind of greenish and the blue. There's a large subunit and a small subunit. And they're separated. And so then, when the RNA comes in, they come together, and then that RNA is going to move through the ribosome. There's actually 3 types of RNA. We're not going to hit that too hard in this class, though. And so the RNA is going to move through, and the ribosome is going to read those codons and put on matching amino acids. So you can see, based on the code, this transfer RNA is going to come in. That amino acid is going to bind to the growing strand. Right? So you add it on, then we shift the RNA over, and we're going to add the next one until we get to the stop codon. Okay? Based on codons. And if you wanted to know what each one was coding for, you would use this. Great. Okay, so let's do a summary slide. So we've got DNA, RNA, proteins. So the first step here, DNA to RNA. DNA is made out of nucleic acids, and the code is ATCG. RNA is nucleic acids, and the code is AUCG, and then amino acids. So we're staying in the same language from DNA to RNA. So we call that transcription. Okay. And then RNA to proteins, we're changing the language. So translation. And this step occurs in the nucleus. And then RNA to proteins at a ribosome. All right. And your RNA is going to get edited before it leaves the nucleus, and then it's going to go find that ribosome, and we're going to do translation. Great. Okay. So now let's hit what happens when things go wrong. So mutations are changes in DNA. One thing to note that they can only be passed on to the offspring if it occurs in our gametes, right? Okay. So if you're functioning now and then you have a mutation in a cell on your shoulder, you can't pass that on because that cell isn't going to be sending DNA into the gametes. Okay. And so there are 2 types of mutations. There's chromosomal, where whole parts of chromosomes are cut or duplicated. Not good. And then there are genes where you get the change of the nucleotide order in DNA. So there's several different options there. And what's really bad is if you make like a stop codon where there's not supposed to be and the DNA stops. But there's also like frame shift where if you have the group of 3 and you take one out, so that's the codon you're reading here and then here. If you take one out, now we read there and then there, and our codon's totally different. You might just have a change in a single amino acid or a single nucleic acid, which sometimes you don't notice because the same code, like CUU and CUC both code for leucine, so changing that last letter doesn't matter. But other times it'll change, and if you change from an amino acid that's like hydrophobic to one that's hydrophilic, that's totally going to change the structure, right? We've looked at sickle cell anemia where just a single amino acid different totally changed the shape of hemoglobin in your red blood cells, right? Okay. And now we have genetic engineering, biotechnology. So that's the field where we're doing research looking at genetics and changing them. Okay. So there's a lot of different applications. I really liked this picture. personalized medicine, you can have genes inserted, more productive fruits, vegetables, increase in GMOs. So, GMO is a bad word, right? But really, there's no consensus that genetically modified organisms cause any harm at all. It depends what you're putting in there, and generally it's not affecting us at all. We can help develop biofuels. They can make microorganisms that eat oil from oil spills, for example, for an environmental biotechnology. We'll help use it to clean our water, sterilize things. But an interesting area of biotechnology is genetic engineering. And the really cool research there is done on CRISPR, clustered, regularly interspaced short palindromic repeats. Sounds really exciting, right? No, it does. It is. It's the coolest thing. Here is a video that's like 15 minutes long on it that's animated and really good. I highly, highly, highly suggest watching it. And CRISPR is basically going to change all of our lives forever. And this is showing how it works. You can insert parts into RNA and make changes. They sell at-home CRISPR kits where people like inject it in their muscles and say, my muscles are going to get bigger. It's a terrible idea to do it that way. Do not recommend that at all. But it's basically, we can be engineered to be better, faster, stronger than we are. It's pretty cool. Please watch the video because it explains it way better than I can. And then gene therapy is another cool area where you can change your genes from mutated versions to healthy working ones. And so there they'll use a virus with the RNA that they want, the DNA, and they'll put that into the nucleus, into your DNA, and then you'll be making the working gene. Pretty awesome. So this is a really short chapter where we are just dipping our toes in the surface. But it is really interesting, and I hope you watch that video on CRISPR, because that's definitely the coolest thing ever. Okay, have a lovely day.